Hello, everyone. Hello, everyone. Thank you for joining us at today's Cactus and Biointron Lunch and Learn. If you did not get a raffle ticket, now would be the time to get one. Once again, if you did not get a raffle ticket, now would be the time to go to the front and get one. All right, so I'm going to give a quick company introduction of Cactus Bio, followed by an introduction of Biointron. Do you have any questions? Hello, can you guys hear me now? Awesome. So I'm going to go ahead and start with a quick company introduction of Cactus Bio. Just take this out. Followed by a quick introduction of Biointron. So in a nutshell, Cactus Bio's mission is to deliver reliable recombinant proteins and GMP enzymes with a commitment to quality, speed, and innovation. So some of the key research areas that we offer solutions in include transmembrane proteins, MHCs and TCRs, cell and gene therapy reagents, as well as GMP enzymes. The first key research area I want to focus on are our transmembrane protein platforms, specifically our VLP and nanodisk platforms. So our VLPs, or virus-like particles, are great for immunization, antibody screening, as well as PK and PKD studies. And then on the left is a nice graphic that demonstrates how our VLP core during the budding process encapsulates these target membrane proteins and surrounds it in the lipid bilayer. So all of our VLPs undergo stringent quality control, validated purity via SEC HPLC. We also validate size homogeneity via DLS or dynamic light scattering. Additionally, we also validate via activity. This could be from ELISA, SPR, BLI, etc. So all of our VLPs have very good stability, and this is a catalog example of our human Claudin 18.2 VLP. As you can see on the bottom, we have stability data supporting nearly 2 years of retained activity when stored properly at -80. On the top left, we demonstrate the retained activity through stability testing over multiple days. Similarly, our VLPs are also stable across multiple freeze-thaw cycles. And then lastly, I did want to add that we do have very good batch-to-batch consistency, as you can see by the curve across 3 different lots on the top right. So our VLPs are able to produce robust immune responses for antibody discovery. Here's an example of one of our popular GPCR5Ds, specifically CXCR4. On the left, you're able to see we get a good immune titer generation, and then by combining HeLa-cell and the CXCR4 VLP anti-serum, as well as HeLa-cell CXCR4 an antibody drug, getting similar positive results. And then lastly, on the right, you can see that we have panels whereby the top and the bottom are positive controls and are non-blocking antibodies, followed by our negative control and then the 2 blocking antibodies. And I'll also add that we can share these slides with everyone, so there's no need to, take scrambled notes or whatnot. So the other platform that we have for transmembrane proteins are our nanodisk platform. These are natural lipid bilayers that display small proteins, typically in the 8 to 16 nanometer range, and these have significant implications in drug discovery, as well as interactions of drug candidates with membrane proteins. We usually see that these are used in applications with binding studies like ELISA, SPR, BLI. So this gives a little overview of how our nanodiscs are produced. We start with target protein expression in HEC293 cells, and through the addition of our SMA-based copolymer proteins, we're able to encapsulate these target as well as non-target proteins in this copolymer. Followed by, traditionally, we use His and nickel column purification in addition to our own proprietary method to stabilize and solubilize these target membrane proteins without the use of detergent. So here's a good example of our biotinylated human GPCR5D nanodisk performing well in two different analytical assays. The first is an ELISA using the talquetamab bispecific antibody, and then we also have good BLI data using an anti-GPCR5D antibody. So just very quickly, we do have a large selection of catalog proteins spanning from single-pass transmembrane proteins like transferrin all the way through to those tricky SLC solute carrier proteins that can span upwards of 14 pass. So the next topic I wanted to jump into are our MHC and TCR solutions. So we offer peptide MHC in a couple different formats. The first of which is an E. coli refolded peptide MHC. These utilize original sequences, not engineered. Typically, what's important to note is that these don't support post-translational modifications, and the production cycle is about 8 to 10 weeks. Additionally, we also offer single-chain trimer peptide MHCs, where a linker is utilized to stabilize the peptide in the HLA and B2M regions. These typically have a single plasmid design with engineered sequences supporting PTMs. So for our peptide-loaded MHCs, we have a variety of different formats, including monomers, tetramers, as well as biotinylated and fluorescently labeled peptide MHCs. So some of the applications of our peptide MHCs include animal immunization, TCR screening and validation, different binding studies, as well as cell sorting. So we're able to demonstrate that we have high binding affinity between our peptide MHCs and the relevant TCR. Additionally, in addition to peptide-loaded MHCs, we also offer a peptide-ready MHC platform. This is essentially a MHC molecule with the antigenic peptide region empty, such that you can use your own custom-produced or custom-sourced peptides and very quickly and rapidly form your loaded peptide MHC. What's notable about this is that unlike other competitors, our peptide-ready MHCs don't require UV or peptide exchange. So some of our peptide-ready MHCs are across different formats, including monomers, tetramers, and as well as fluorescently labeled and biotinylated versions. So I think it's important to note that our loaded peptide-ready MHCs have near equal performance to our preloaded peptide MHCs. As you can see, we have an example of a peptide-loaded single-chain trimer monomer compared to a loaded peptide, and then again, we have the MHC with no peptide present as a negative control. So additionally, our peptide-ready MHCs we've found are able to bind to both affinity-matured TCRs, as is demonstrated by the control peptide with no binding, and then the loaded peptide with good SPR data on the right. Similarly, we find our peptide-ready MHCs are also good at binding to low-affinity TCRs. So the next thing I wanted to talk about is our soluble TCR expression services. We're able to offer different formats, including SCFV TCR, TCRs with HIS or other tags, and we're able to engineer these TCRs based on both predictive TCR modeling and our past experience. SPR and ELISA is a very frequently requested form of QC, and these are often integrated as standard QC for our TCR expression services. And then just quickly on the right, I wanted to show that all of these are catalog items, but we also support custom production if, maybe our catalog item isn't exactly how you need it, or maybe it's a brand new target that you don't see. So I also wanted to talk about our cell and gene therapy reagents. So we do have a plethora of off-the-shelf cytokines at both GMP and GMP-ready grade. The main difference here being our GMP-ready already have an established protocol in production, and we have a GMP facility that is not at capacity. And, if your company is looking to transition from preclinical to clinical, this is a great opportunity to transition some of your research grade reagents into more clinical applicable GMP. So we also offer AAV titration ELISA kits. These are able to quantify both full and empty intact AAV capsids We cover a variety of different serotopes with no serotope cross-reactivity. These have high sensitivity and precision and offer a wide linear range. And then on the right is just an optimized protocol that we usually show in our ELISA kits. So we do offer high batch-to-batch consistency and specificity across AAV capsids. So as you can see on the left, we have two different lots of our AAV9 showing near identical curves. And then on the right, we have a good graph showing the specificity of our AAV9 kit to intact capsids as opposed to denatured. So our AAV kits also have very high specificity to the specific AAV serotypes. So for example, on the left, you can see our AAV9 kit doesn't have any cross-reactivity with AAV2, 5, 8. And then similarly, on the right, you can see our AAV2 kit doesn't have any cross-reactivity with AAV3. So if you don't see the AAV kit that you're looking for, we also offer custom proprietary immunization schemes where we often see we're able to overcome weak AAV immunogenicity. Sorry. And lastly, I just wanted to briefly talk about our GMP enzyme offerings. So we offer a max nuclease. This is a non-specific endonuclease with very high activity. It degrades all forms of DNA, RNA, including single, double-stranded, linear, as well as circular nucleic acids. And then on the right, you can see this PCR product, as well as genomic DNA and plasmid DNA shows comparable degradation compared to other competitors. So some of the applications that we see these max nucleases used in include viral vaccine and viral vector purification, the removal of nucleic acid residues in biological products, as well as reducing the viscosity of cell lysates and cell supernatant. Sample preparation can also be used with this max nuclease to improve Western blot, electrophoresis, ELISA, as well as various chromatographies. And then just quickly on the right, we have a little graph that kind of shows the different reaction conditions from which the max nuclease either retains or loses much of its activity. And then really quickly, I just wanted to show that we do have very stable max nuclease in that we're able to see a retention of activity across high temperatures, as well as multiple freeze-thaw cycles. So in addition to the max nuclease itself, we also offer max nuclease ELISA kits. These are great for detecting and quantifying residual max nuclease with both high sensitivity and specificity. So here you can see an example of the sandwich ELISA that our ELISA kit utilizes, and then an example curve showing concentration. So we also offer various DNA and RNA synthesis enzymes, starting from your template preparation all the way through synthesis, capping, cyclization, as well as tailing, and we support both custom services as well as the individual enzymes for your own use in in-house IVT. So our Max Pure does have very good yield integrity, and it reduces the amount of double-stranded DNA, especially compared to our first generation T7 RNA polymerase. So our Max Pure polymerase is also very stable across different temperatures and multiple freeze-thaw cycles. And lastly, I just wanted to add that we have a newly launched ultra-low endotoxin protein line. This is going to include not only catalog products that we are performing an additional EU purification on, but also custom requests where the application is some in vivo work, and we really want to keep that EU down. We can pretty regularly get to at or below 1 EU per milligram by the LAL method. And again, thank you for your time, and I can share these slides with everyone afterwards. So thank you, and I'll go ahead and introduce Hao Ming. And if anyone has any questions, feel free to ask.
The single chain PCR, if I remember correctly, engineering single chain PCR can be quite a difficult task. Wondering if you guys only do single chain PCR production using customer provided systems, or you have capability for developing your own single chain PCR?
We typically rely on our own engineered TCR sequence, but we do have experience using customer provided ones as well. Yeah, I can follow up with you after. Any other questions? Awesome, thank you.
Hello, everyone. All right. My name is Hao Ming. I'm the Senior BD Manager with Biointron, and I will give you a brief introduction of our company. Some of you might already know, but for those people who do not know us, we are a one-stop shop antibody CRO, provide preclinical antibody services. So you can see we have more than 10 years experience on antibody services. We have more than 600 employees all over the world. You can see our production facilities in Taizhou, Jiangsu Province, and our headquarters and R&D center is in Shanghai. We also have a logistics center in Netherlands, Europe, to help support our Europe branches, customers, and our office is in New Jersey, and we recently have an office in Harvard, near Harvard. So we have supported more than 2,500 customers globally, and we're also an ISO 901 certificate company with this 2015 version. Here are some of our photos of our production facilities. You can see the clean rooms, the sequencing machines, automated working stations, and the shakers, bioreactors. We have the capability to handle even 200-liter expression for your materials. So people always ask how Biointron can produce antibody just within 2 weeks. So we not only optimize all the process, but we also support it by these automated working stations to help us accelerate the production as well as reduce the human mistake. Here are some photos of our Shanghai R&D facilities. You can see we're fully equipped with our experienced scientists. So what does Biointron do? Firstly, we do antibody discovery. We currently have 2 platforms. One is phage display-based VHH antibody discovery platform by immunizing alpaca. Another one is a microfluidic-based single B-cell screening platform by immunized mouse or rabbit. I will elaborate it in the following slides. And for the antibody protein productions, we're using transient expression in CHO hepatitis 3. We can also help you generate stable cell line for your long-term antibody production. And for the antibody optimization services, we can conduct humanization and affinity maturation. For the cell line development, we currently offer overexpressed cell line generation, which can be used for your future assays or just for immunization. For the CHO-K1 cell line production, R&D use-based stable cell line generation, we can help you do that. We can also sub-license our CHO-K1 BN cell line for you commercially use originally from ECACC. For the in vitro assays, like SPRs, BLI, FX, they can be performed with us. We can also help you do some functional assays like internalization, T-cell activations, extra. They are always associated with our discovery campaign to do the to the binder validation and characterization. We also have some catalog products like biosimilars and isotype controls. They're already on shelf and ready to be shipped. So for antibody expression, Biointron is the first company who have this concept of 2 weeks antibody production from gene synthesis to the final QC, which only takes 2 months. 2 weeks. And for the international shipment from China to US, it takes 1 additional week. We are able to handle from 100 microgram to even 100-gram level production, and we have the capabilities to produce more than 1,000 antibody per batch. The buffer can be customized or using default ones. We deliver not only the purified antibodies in the report, but also the expression plots that contain your gene of interest. No matter you are looking for small-scale screening, we have those specific packages like rush maps, which can be smaller as 100 microgram as an amount guaranteed delivery. But also we also have some isocollected antibody production if this is something you're interested in. And for the production size, you can start from 30 milliliter to even several liter level. And if you're satisfied with our services, we can always help you do the upscale production. So for the large scale production, all the antibodies will come from one batch to maintain the consistency. Endotoxin level can be less than 0.1 U per milligram, which for those large amount, people always use that for the animal studies. So the turnaround time will be fast, as short as 4 weeks for gram level production, and we are able to handle more than 100 grams level production as well. Any questions on the productions wise?
For the protein QC, how much protein do you load for QC?
For
For HGL, 5cc, what's the amount of the antibody you load?
this depends on like how much.
You can load less, maybe you can load more, maybe other bands show up. Any standard? You have some 2 microgram loading or 10 microgram loading?
This I need to check. Yeah. Sorry. All right. Other questions?
Cell line production, you are doing research pool, you like monoclonal stable cell line.
We can do cell pool, which we can also help to generate the stable cell line, which is originally from one single clone.
Do you do knockout? Not yet.
Not yet. Yeah. All right. Okay, let's go next for the antibody discovery. So I will introduce the VHH antibody discovery first. So you must have noticed that we have put alpacas in your goodie bag. So we have a farm in China which have more than 300 naive alpaca. And yeah, we use that one alpaca for one project, which will eliminate the background interfere. All the alpaca have a traceable health and immunization record, which could reduce the mix. And we can deliver you a certain number of unique binders at the end of the discovery campaign. I'm sure everyone is quite familiar with this, but we start from alpaca immunization, hematopoietic connections, the iron extractions, and library construction. After that, with phage display and biopanning, we're able to acquire the positive clones and a positive clone sequence. And with the sequencing, we will get the VHH sequences. And with that recombinant produced, we can do more characterization and validation. I didn't put much case studies, but this is one thing that people mostly focus on is the binders affinity, which these are from more than 600 SPR results come from 30 projects from one of our customers, which have the different targets. But you can see for the affinity level, they're mainly from 10 to the negative 8 to 10 to the negative 9. Yeah. Any questions on VHH part? Cool. I will move forward to the microfluidic-based single B cell discovery platform, which we call that AB drop. So for the immunization, we can start from DNA, RNA, or LNP, or anything KACTUS produce for you. And we also use our express online or just soluble proteins for immunization. By immunizing mouse or rabbit or other transgenic mouse, after 1 to 2 months immunization period, we will harvest plasma B cells from bone marrow or spleen, and then perform the single B cell screening. With this part, we can significantly reduce the time comparing to the traditional hybridoma process for like 2 to 3 months time saving. So you can see this throughput is like million level plasma B cells can be screened, and then we can collect those positive ones with the Fred methods, and with the positive clone sorted, we're able to perform the NGS and getting those natural paired sequences for future production. The advantage of this platform is that for only 1 day, we can screen millions level of plasma B cells, and for the NGS and sequencing data analysis, it takes 1 week, and all the stuff has only need to be conducted once, and so we have all the information for future. And from screening to purified candidates, it takes around 1 month. And for reference, the total discovery campaign will take 2.5 months to 3 months. That will be all. Is there any questions for this platform? Okay, good. Okay, so here are some of our current collaborators like AstraZeneca, Sanofi, and NIH also working with us, as well as some Harvard Biomed and Sagar. And hopefully, like globally, we have more than again, we have more than 2,500 customers collaborating with us. And if you are looking for more information, please feel free to speak to me or my colleagues, Huan Yi. We'll be around during the networking area. And thank you everyone for the time. Right. So we can skip the networking and enjoy the lunch if you arrive a little bit later. Yeah. Hello. Hello, everyone. Yeah, we're about to have the talks, and hope everyone enjoy the food and the networking. So I would like to introduce Dr. Sagar. So he's the senior principal scientist at Sanofi, and he will bring us a talk about the antibody developability. And welcome. Thank you.
Okay. All right. Thanks. Can you hear me? Yeah. Okay. So I work just a few blocks over to the east, but this is my first time over here. It's a really nice location. I didn't even know it existed. So happy to be here. Can't hear me? Better? Okay. All right. So I think I kind of have to give an explanation for my topic. Or the title of my topic. It's honeybees or butterflies. I think developability, I think in the industry has become kind of a bad word. We've become these butterflies that are, one way of pollinating a garden, or we could be honeybees too. We both have the same purpose. You pollinate a garden, but you have butterflies that lay eggs on your vegetables, and then you know what happens to them. Whereas with honeybees, you can actually get a second product, which is honey. So, there are two modes of looking at the same thing. And I'm going to talk a little bit about how we're transitioning, or we're trying to transition from this honeybee mode, the butterfly mode into the honeybee mode. And I think if there's one thing that I want you guys to take away from this is that that's a lot of hard work. So, buzzing as a bee is because that's hard work. Before I continue, I want to thank my collaborators who are actually just as much authors on this work as I am. Uh, I'll point out a few of my team members. Shahua, he's done a lot of the proteomics work, that proteomics-based peptide mapping that I'm going to talk about. Samantha was also in my team, and she's done a fantastic job of handling a lot of the actual samples that we go through. And the other folks are actually collaborators with us in different parts of Sanofi, in different countries. So definitely not a one single site work that I'm going to talk about. We have collaborators from Germany, we have collaborators from France, and we even have a collaborator from Gene Data Biologics, that's on Brandenburg. In Sanofi, we've had this paradigm of developability for a while, where you go from let's see if this works. Nope. You go from lead identification to lead optimization, and then you go into your pre-candidate selection. So both the research organization and the CMC organization have come together to come up with this early research concept where we address issues that we can identify in the beginning, in the middle, and towards the end. And that's essentially how we've broken it down looking at the actual variable domains, what's happening to them, understand the liabilities that are associated with that. Once we've optimized those liabilities, we try to look at the molecule in its entirety and try to fix that molecule if there are problems that we've identified. If we are unable to, then the problems will be translated back towards CMC, and then it becomes a process issue. So that's where we start to look at it more from a process perspective, and address issues that we were not able to solve in research. We just pass that ball on to the CMC folks. I think one thing that we need to understand, like I said, it's a bad word these days, but what does it actually mean? It's really, it's ubiquitously used in not just our industry, but it's essentially, can you take a specific molecule or product to the next stage? That's what is essentially developability. What our function in early research is to identify risk, present it to a committee of people who will actually decide, okay, what is the acceptable amount of risk, and take that forward and decide what's going to be our candidate going forward. And the reason to do this in early research is that you can actually take some of the burden away from from the development folks, right? So if you're able to invest a little bit of time and effort into in preclinical research, you can actually reduce the burden, but not only reduce the burden, you can actually bring the product to market sooner and have a better product. So that's why we do this early investing in quality. The outline of my talk, I'll just spoke about the why. I'll talk a little bit of how we do it and what we do. Talk a little bit about how that translates, and then go into what we're doing for making it better. And like I said, if there's one thing that I want you to take away, the idea of minimalism is a lot of hard work. Yeah, it doesn't, it's not an easy task that we've given to ourselves. There are these 4 pillars that I will talk about that we consider for developability. And this is manufacturing, which basically tells you about how much you're going to have, the product that you're going to have, how pure it is, what's the molecular integrity. Then you have stability, which can be either chemical liabilities. You can think of physical instabilities. We want to understand those. On the clinical side, what's happening is that binding to something off target, other degradation pathways within in vivo, and what's the PK. And then finally, deviceability, which is now very important to us, as a lot of the drugs are actually on you want them to be in self-injectors and things of that sort. So you want to be able to get that to high concentration without any viscosity, opalescence, or interactions with the device. We do this in an integrated way in our research platform. Right from molecule, we keep developability front and center, right from molecular biology, protein production, purification, and all the way into the analytics. So the idea is always to have, your best molecule going into development. I won't go into details over here, but just to get on with the talk. I think this is something that almost every person here would be well aware of. At the beginning of our campaigns, we have hundreds of thousands of molecules that we're expressing, and they're usually not very high purity. A lot of molecules, very low purity. And as we go through the value chain, we end up with the lead candidate, which we can actually produce in a large quantity and very high quality protein. So when we are trying to do work in developability, we're trying to understand not the batch, but the protein. And that becomes more and more difficult with the idea of, impure proteins. You're only looking at the quality of the batch. You're not looking at the quality of the protein. So trying to understand the distinction between them is important, and to be able to have readouts that are actually independent of purity is important. that's for a simple monospecific. What happens when you put it into a multispecific, like over here in this case, there's a bispecific protein that we're generating. You, of course, have the same problems for the two sets of monospecifics that you're going to put into the bispecific. But then now you also have the problem of, can that data actually translate, right? You've made a bispecific, do the properties of the monospecific translate to the bispecific? And a lot of times the answer is no. So identifying those pain points is important. And here's one example. I think Sanofi is pretty famous for our CODV molecules, which I'm showing here on the right. Well, I guess, right, yeah. My left. Anyway. Yeah, and you know, I've listed out all the issues that go with making a more complex molecule. Uh, for sure, you know, you have the advantage of being able to bind more number of targets, but then you end up with a lot of other problems with that protein. Here's one example of what we can expect to encounter. Simple bispecific, you can end up with so many different combinations of product. And there's a bunch of different ways you can try to identify them. All of these different assays are something that we would do typically, but you can imagine running all of this for thousands of molecules can be a huge task and not something that you really want on a day-to-day basis to be dealing with. All right, so I'll talk a little bit about what we've been able to do in being able to predict accurately very early in our discovery stages. And I think the tick marks seem to have moved between PC and Mac, but I'm really pointing to the check marks are supposed to go to the chemical stability, and this darker check mark is going to the colloidal stability. The lighter blue is really on the conformational stability. We're not really talking about physical stability today. I'm going to talk about the chemical and colloidal stability. We have had good success with conformational stability, but that's, I think, of lesser interest to us in the antibody world. I think it's more important for people working in nanobodies and in maybe more fragments of antibodies like scFvs and things of that sort. So I'll go forward. Why do we care about chemical liabilities? These are our top pain points that we have with chemical liabilities. You can have aspartic acid isomerization, you can have asparagine deamidation, you can have methionine oxidation or tryptophan oxidation, and these can actually influence the structure and function of the protein. You could have a simple thing where you lose function, nothing else happens. You've just lost some amount of your protein. That's the simplest outcome. Where you can actually even have worse outcomes than that, where you know, you end up with aggregation. You can end up with immunogenicity, which is probably your worst nightmare. And of course, everything that goes with a chemical modification, fragmentation, and structure disruption. So that's actually why we care. What do we do about it? Typically, we would address this issue at the point where we've selected our top molecules, right? So you're around where those 4 or 5 molecules are left behind in our bucket, and then we're saying, okay, which one of these does not have chemical liabilities? Take that forward. But that means that we're going to actually wait until the functional work is all completed and then say, okay, now we start the developability. Wait another 2 months before you make your selection. And we definitely don't want to do that. What we want to be able to do is start doing the chemical liability detection work right here, where you have the hundreds of thousands of molecules. By the time they've decided which one of them is functional, I should already have the data that says something is clean or not clean. And that's really the goal over here. Can we take that and make that happen? This is where Xiaohua's brilliance comes in. Many times in the past, we've run tri-specific antibodies in our peptide mapping workflows. And he says, well, if I can do this with tri-specifics, right, where I'm saying, okay, this peptide belongs to this particular variable domain and not to that variable domain, why can't I just do it with tens of antibodies, right? As long as the CDRs are not the same, I should be able to identify where the chemical liability, which molecule the chemical liability belongs to. So we collaborated with our colleagues in France. We came up with an algorithm where we could select only the molecules that have distinct CDRs and put them into our peptide mapping and workflows, where we identify the liabilities. And here's a proof of concept that we showed with commercial antibodies that we had sitting around in the freezer. You try to stress the molecule, identify the liability on each individual one at a time, mix them all together into one sample and do the same experiment again. And you can actually see over here that we've got pretty good correlation. We can identify the modifications to the same extent in either experiment. So that gave us some hope. And like I said, we came up with this strategy along with our collaborators, where we basically had this algorithm developed where we can identify all the sequences that are closely related and make sure that, if let's say you have a green family, a red family, and a blue family, don't make a sample with all reds, right? Have sample with one green, one red, one blue, and use that set of molecules to mix together. We've got all of our workflows automated so that we can actually just then use the output from this algorithm, apply it to our workflow, and go straight into our LCMS workflows. We've been able to reduce the amount of experimental work that we do, but then we want to also be able to take that experimental data, put it into the software that can then spit out, exactly what we want to know. So that's where we collaborated with Gene Data Expressionist, and we've made this into a semi-automated workflow where we can actually then the software gives us a list of peaks that we actually need to monitor. The rest of them get auto assigned, and it just goes ahead. So here's what we started with. You had a list of all putative sites at which you could have modifications. We end up with whether or not there is a modification on that site. This is a huge help in our next step, right? Which is essentially saying, by the time the functional data is collected and ready, and I've selected my top 10 molecules, 20 molecules, whatever it is, I also know whether there is a developability cost to it. So there is no waiting needed. We're on the same timeline as the functional assays. Makes a big difference in deciding whether I'm going to engineer something or just go and pick something else right away, because I have very similar function in that. Second part that I'm going to talk about, how are we doing on time? Okay. Second part I'm talking about is colloidal stability. So this is something that, like I was talking about, do characteristics of monospecifics translate to multispecifics. Colloidal stability is one which will never do that. Yeah, you will never be able to take the measurement of monospecifics and say, okay, I know what's going to happen to the multispecific. Maybe at some point AI is going to step in and say, prove me wrong. But for now, as long as we're measuring it, it is not true. So this is where we want to then step back in and think about this a little bit more. So what is it that really drives things like viscosity, for instance, right? So usually it's not the constant domain. It's always the variable domain that's driving your issues with viscosity. And that's because you're going to have some kind of electrostatic dipole, and that's actually made worse with multispecific antibodies. And I've quoted a well-cited paper over here, so you can go and look into why that actually happens. The more complex the molecule, the more diverse the arms are going to be, the more difficult it's going to be for you to up concentrate it and have no issues and be able to put it into a device. And that's always true. So this is another paper that's published by my colleague John Kingsbury in Sanofi. And what he's showing here is that you have a measure called KDDLS, which can predict very well opalescence and viscosity of proteins, of antibodies. While there is no, excuse me, it's not a direct correlation, you can actually use a cutoff value. Like in our case, what we do is we use a cutoff value of KD of 20. Anything below that we consider to be high risk. Anything above that we consider that to be low risk. Now that's an assay that we can do at low concentrations. Viscosity, typically we would be measuring at 150 mg per mil. This assay can be done between 5 and 10 mg per mil. Big improvement, but that's not good enough for us, right? We can't do 5 to 10 mg per mil when you're doing thousands of molecules. You need something even better than that. And so this is another one out of the same lab, John Kingsbury's lab. This time it was authored by Charlie Starr. And what we've got is a similar assay, CS-SINS, which is a very low concentration solution that you're using to predict high concentration behavior. And I've quoted the paper, so you can actually go and read it more about it. The one issue that it does have is that it doesn't really work very well with IgG4s. It's great for IgG1 assessments. And what we've been able to do with this, then the paper as it has been presented works very well with very low concentrations, but it does not work well in high throughput. So we've actually taken that assay and developed it into a higher throughput assay, and a paper is being published on that in Jove. So please watch out for that. And I'll just skip to the end of it. I won't take away the my colleague Marissa's work. I'll just jump to the end point where I can say that we've successfully started implementing the CS-SINS assay the moment the bispecifics or multispecifics are generated. Thousands of them, we can run this assay and actually use that to predict solution behavior. So hopefully I've convinced you that at least two of the most critical assays that we consider in Sanofi for developability, one is chemical liability and the other is colloidal stability. We're actually able to bring them up much earlier in the process and make it so that we're ready with those answers the moment the teams are ready to select their top molecules, reducing the burden on engineers and reducing the burden on the timeline for developability. And our next stage that we're trying to handle are the manufacturability packages and the physical stability packages. That's our next butterfly that we need to tackle. So I think with that, I'll end my talk and take any questions. Yeah.
So as in nowadays you can use very fundamental tools to look at the amino acid sequence to see what's the potential variability is. And do you see a good correlation between this prediction with the real-time data?
Yeah. So we've actually, obviously, we're generating data on thousands of molecules here. So the next step really is to take it into an AI model where we can actually then just, stop doing this work and just predict it. I can't discuss those details, obviously, but I can say that we're close. Yeah. We should be able to predict that pretty soon. Yeah. All right. I guess that's all the questions. Thank you.
Please give a warm welcome for Alex, our next speaker.
No matter how hard I try, I will not top the raffle excitement. So, all right, that's me. I'm Alex. Okay. So I recently joined the KACTUS team, but Jordan already gave a great intro to what KACTUS does and the recombinant protein technologies we use. So I decided to give a talk on something more, I don't know, fun, I think. This is actually the final chapter of my thesis. Um, and if you're in the translational medicine field, you know that the final chapter is always like that sandbox, didn't finish, won't publish ever, fun chapter. So hopefully this will be interesting. And also, when you ask yourself through this presentation, why did he do that? The question is, I don't know, because I could and I had time. So, well, might be more nuanced than that. But so the title is Driving Towards Endogenous Immunity in a Rental Car, because everyone who does CAR T cell work has to come up with a crazy title that doesn't really mean much, but sounds cool. Okay, so first I want to introduce you to Dr. Coley, if you don't already know him. So Dr. Coley was one of the first physicians and researchers to understand how the immune system was involved in cancer therapy. Um, so what he did was he took a couple different species of bacteria and injected them straight into tumors and realized, or at least the legend goes, that he cured hundreds of patients by injecting his toxins directly into the tumors. And this led people to question, like, how is it that when you kind of infect someone, their tumors go away? And people realized that, well, maybe he's kind of labeling these cancer cells that are our own tissue as a foreign substance. So your immune system recognizes it as an infection itself. So this gets into a little bit of basic immunology, viewing cancer as a pathogen versus viewing it as a wound. So immune cells have different receptors on them called pattern recognition receptors. And these are kind of split into two categories. So there are pathogen-associated molecular patterns called PAMPs and damage-associated molecular patterns called DAMPs. And these PRRs recognize either PAMPs or DAMPs. So PAMPs come from something that's outside of the body, either viruses or bacteria. Molecules like LPS or viral DNA, flagellin, these types of molecules tell our immune system that there is something invading our body and that you need to attack it and kill it. However, what a tumor does is it helps release PAMPs into your, DAMPs into your system, telling your immune system this is damaged tissue that needs healing. So things that are released from our own cells that shouldn't generally be in the blood or the lymphatic system, but they are indicating that there's cellular damage, such as ATP, heat shock proteins, mitochondrial DNA, etc. So these different DAMPs send different signals to your immune system, recruiting things like tumor-associated macrophages, Tregs, myeloid-derived suppressor cells to help kind of put the immune system at bay and help that tissue regeneration with blood vessel formation, a dense granular tissue kind of shell around the area, as well as a lot of anti-inflammatory cytokines to help, again, suppress the inflammatory immune system. Our goal, though, is to avoid the immune system receiving all these signals that are being sent by the tumor and instead redirect it towards a infected type of system so that the immune system attacks and kills the cancer. So the question is, what is self-derived that can make the cancer cells look like non-self? And the answer to that is mutations. So we kind of set up this battle here of how can we harbor these mutations to outweigh all of these signals that the cancer is sending towards the immune system to let it grow. So how do immune cells seek and identify these mutations? So let me give you a brief intro into some of the basic immune cells that you probably already know about. So T cells survey and look for non-self inside of the body with the help of dendritic cells. So cell debris is uptaken by dendritic cells and then presented to T cells. And the T cells are built to identify what isn't already part of the body. So when the dendritic cell and the T cell come together into an immune synapse, what happens is a few signals are transferred that are needed to activate the T cells. The first is the peptide MHC combines with the T cell receptor. Signal 2 is co-stimulatory or co-stimulation via co-stimulatory ligands and receptors. And then the third is stimulatory cytokines secreted from the dendritic cell to the T cell. And then with all these signals being met, the T cells are activated. They can go into the lymph node, proliferate, and then directly kill any cells that have these antigens that were presented to them. What the tumor does is it tries to completely stop this process from happening. Now, let me introduce you to B cells briefly. As you all know, this is an antibody series, so you all know about antibodies and B cells. But when a B cell is activated, it can turn into a plasma cell, and then the plasma cell kind of secretes the B cell receptors as antibodies to circulate throughout the body. And these antibodies bind to antigens via simply a 3D surface. So there's no need for a peptide to be woven into an MHC to go to a specific TCR receptor. It can just identify a three-dimensional surface. And just as an example, this is the HIV envelope protein, giving different examples of domains that are kept throughout mutation to create antibodies that can bind, called broadly neutralizing antibodies. Okay, so now we get to the CAR. What is a CAR? A chimeric antigen receptor developed 25-ish years ago now and perfected about 15 years ago takes the binding capacity of the B cell receptor and combines it with the intracellular signaling mechanisms of the co-stimulation as well as the TCR. And you get the CAR T cell. So because you have the intracellular signaling mechanisms, you have the cytotoxic capacity of a T cell combined with the surface antigen recognition of a B cell. And with that, we cured cancer. Not actually. Well, sort of. When we look into how this was translated into the clinic, we found that the first CAR T cells that were approved by the FDA were for CD19 leukemias and lymphomas. And they actually worked great. So it was kind of interesting. The first one that was approved worked very well. Gave up to 80% cures for different leukemias and lymphomas. But we haven't seen that same success translate into solid tumors. And a recent review paper shows that in all different types of clinical trials for solid tumors, we often see zero results at all, any objective results, partial remission, complete remission, nothing. So there's really a struggle to develop this CAR T cell therapy for solid tumors. So researchers have tried to farther engineer the CAR T cells to better overcome the obstacles that solid tumors present that the blood cancers don't. So to do this, they've taken many approaches. Dominant negative switch receptors to try to trick the T cell into seeing something that it doesn't. Gene knockout models, trying to enhance the T cell metabolism to survive better and overcome the immune suppression. One of the most potent ways that people engineer CAR T cells, though, is with cytokines. And this is the approach that I wanted to take. So people have tried different types of cytokine engineering for CAR T cells, but they seem to fall short every time, whether they do an inducible soluble cytokine, transmembrane cytokines, inducible transmembrane cytokines. There always seems to be something that falls short, and most of the time it has to do with the toxicity of cytokines because they're extremely potent. So the problem here is there's a very narrow therapeutic index with the CAR T cells that are engineered with cytokines. The idea being that when you have a high enough dose of the CAR T cells to show efficacy, that also introduces toxicity. So if we can somehow separate those two curves so that we can have efficacy without that toxicity, then we'll have that successful translation into solid tumors. Okay. So this is where you're going to be like, what's he doing here? So to have some fun with this, I wanted to look at what the difference is between um, the longevity of the expression of whatever cytokine I choose to engineer into these CAR T cells. So I was looking at both standard mRNA coming off of the COVID high and looking at something that was a little more nuanced, like a self-replicating RNA, or a replicon as we call it. So everyone knows that mRNA, you deliver the transcript into the cell, and it's read and it produces one of the payload proteins. They last for a few hours, maybe even up to a few days. With self-replicating mRNA, what it does is it takes the replicase from an alpha virus. So it's the non-structural proteins, just what replicates the viral DNA or viral RNA. The replicon is read, it makes that replicase complex. And what that does is it rereads the replicon, it creates a negative strand. From that negative strand, you create the original replicon plus all the subgenomic transcripts. So from one replicon, you get a reversion of itself plus a bunch of proteins of the payload from the subgenomic promoter. The idea with this is that the self-replicating RNA, instead of lasting from a few hours to a few days, it lasts from a few days to a few weeks and expresses at a much higher concentration. So if you're looking at, if you run these cells with replicons through flow cytometry, you'll see the MFI is just much higher. Okay, so one of my collaborators, who's now a professor and moved back to China, he helped develop the delivery mechanism of these replicons into primary T cells, which if you've ever worked with primary T cells, you know that this is a humongous feat in and of itself. So after he developed this liposomal type product, he found that if you did a lipoplex formation, that is coating the surface of the liposome with the RNA material, it transfects much better than if you actually encapsulate the replicon inside the liposome. We found that there is very little toxicity in the transfection process, getting the replicon into the T cells. And then we see here that increased longevity of the expression of the payload. So it looks here like, yes, it is expressing longer, but it still seems to be a lower percentage. But when you look at that MFI again, it's orders of magnitude higher than what just an RNA can produce. So our next step here is to figure out what payload we want to deliver, what cytokine is most critical in helping that CAR T cell do what it needs to do inside of the solid tumor environment. So we evaluated some of the standard T cell related cytokines that people tend to work with here, IL-2, IL-15, IL-12. And then evaluated the different parameters of what issues are being run into with CAR T cells in solid tumors. And we found that there was kind of a back and forth between should we use IL-15, should we use IL-12? They're both good, but for different reasons. So we pursued and just tried out both of them. So one more twist that I want to put into this that brings us into antibody territory is that we didn't just work with the soluble cytokine. Instead, we set up a platform where once we transfect the CAR T cells with the replicon RNA, we actually have the cytokines tethered to an anti-CD45 nanobody. The idea with this is when it's secreted, it can bind to the CAR T cell, so it's anchored to the membrane. But because it's CD45, it can also anchor to any nearby T cells that weren't transfected or any other immune cells and can help proliferate that process. But again, it's self-replicating RNA, so it won't last forever. The idea is we kind of give it a big punch initially. It anchors to the membrane, so it lasts a little bit longer, and then will eventually go away once it has that initial boost. So we tried this concept with both IL-15 superagonist as well as an IL-12. Okay, so expressing these molecules in T cells and looking for the longevity of this expression, we found that there was a different timeline of how long the T cells express the construct. which it was very interesting to us because the size of these two constructs is very similar. So there's something either going on with the actual expression of the self-replicating RNA molecule or with the construct itself and how long it's able to be secreted. We weren't able to figure that part out, but so what we have right now is an IL-15 molecule that doesn't actually express that long. By day 7, we start to see it get off of the surface and it's not there anymore. With the IL-12, it lasts at least 2 weeks and a little bit longer as well. This isn't either good or bad. Maybe the shorter expression is better. Maybe the longer expression is better for IL-12 or IL-15. So, and that's just another way to visualize how long the IL-12 is able to express and stay on the surface there. Okay, so we took these in vivo to test them in a CT2A glioma model of the mouse. So this is a syngeneic model, so we can get the full immune response going with this cancer therapy. So we let these glioma tumors grow in a flank model for 10 days, and then we lymphodepleted as is standard, and then infused the CAR T cells, both the IL-15 construct and the IL-12 CAR T cells. We saw that for the expression, we got about 40% positive CAR T cell transduction with the standard viral transduction, and then we're able to get 100% transfection with our lipoplex system. Okay, very shockingly, we found that the IL-12 CAR T cells very quickly cured all of these tumors, and with even more shockingly, very little toxicity. So something about this combination of technologies is allowing the cancer to very quickly be killed off by these CAR T cells. So we looked farther into this. So these are the individual tumor growth curves. So one of the tumors was cured with the IL-15 replicon, but we decided to proceed with the IL-12 replicon because the results were so astounding. So we took these mice that were all cured and decided to look farther into what was going on in the immune system. Here's a number of plots here, but this is what I want to highlight is I've divided these rows between the CAR T cells, the infused CAR negative cells, and the endogenous T cells. So first and foremost, we found that the first box here shows that the IL-12 construct does not change how much the actual CAR is expressing on the T cell surface. So that's good that the IL-12 doesn't impact that. For the infused CAR negative T cell population, so this is cells that were infused that did express the IL-12 replicon, but did not successfully be transduced with the CAR. We found that there was a decrease of IL-2 and an increase of inflammatory cytokines. So something's going on there. Even though they don't have the CAR, they're still being influenced by the IL-12 to secrete more inflammatory cytokines. And then for the endogenous CD3 T cells, we see that same increase of inflammatory cytokines, meaning that just by transferring these cells into the mouse, it's training the endogenous immune system to be more inflammatory through the IL-12 cytokine expression. Okay, so we have a lot of different parameters going on here that we're engineering this CAR T cell with. We want to find out, are all these different modifications actually necessary? So we designed a whole bunch of groups to test this. Is the CAR important? Can we just have T cells that express the IL-12? Is the anchoring to CD45 important? So we swap the anti-CD45 with an anti-mCherry. Is anchoring to CD45 better than just doing a transmembrane IL-12? Is the replicon RNA actually better than just using mRNA? And finally, is the transient expression even necessary? Can we just have it constitutively secrete the anti-CD45 IL-12? So we created all these constructs and we tested their activity with a pStat4 assay. And sure enough, all these constructs with the IL-12 had active IL-12. Okay, so we put all of these in mouse models. And again, we're kind of blown away to find that indeed this specific combination was necessary to get the results that we saw before. So this time we saw many cures. And in the top right there, when we were looking at toxicity, you can see that one plummeted in body weight, and that toxicity came from the constitutively active, constitutively expressing IL-12 construct. So we do indeed need that transient expression to keep the toxicity low. All right, we wanted to step things up and do a harder model than just the glioma. So we used a B16 F10 aggressive metastatic melanoma model and an anti-TRP1 CAR T cell. So we let these tumors grow out for 5 days, lymphodepleted, and then transferred the CAR T cells into the model. Similarly, we saw that compared to standard CAR T cells, the IL-12 replicon CAR T cells significantly suppressed the tumor growth and increased survival. Okay, so what we're really interested in is how does the IL-12 being secreted from these CAR T cells influence the endogenous immune system? Because it's very well known in the cancer immunotherapy world that you need to recruit as many endogenous T cells that can identify as many different neoantigens as possible to keep the cancer at bay and to prevent or to overcome antigen loss and attack it from many different directions as possible. So we set up this experiment, again with the glioma model, where we came in after a week after treatment and phenotyped the entire immune system, as well as ran an ELISpot. And for those who are unfamiliar with an ELISpot, it's very similar to an ELISA, except it's with the cells and not just the serum. So what we do is we harvest the T cells from the mouse and incubate them with antigen negative cancer line. So the idea is that the CAR T cells recognize I guess our target here is EGFRV3. The CAR T cells recognize our mutated cancer antigen, but we're going to use an EGFRV3 negative cancer line. That means if there's any activity, any T cell activity, they're being activated by a different antigen than our engineered cells were, meaning that there's some sort of antigen spreading going or epitope spreading. Our endogenous immune system is starting to recognize different mutations in the cancer than we engineered it to. Okay, when we reached a week after the therapy transfer, the first thing we noticed is that the spleens in our treatment group of import were significantly larger, meaning that there was more T cell proliferation going on and more mass of T cells in there, which was a good sign initially. That meant more activities going on in the spleen. Okay, so looking at phenotyping inside of the tumor, we found initially that there were significantly fewer antigen-presenting cells inside of the tumor with our treatment group. This was a bit shocking to us. We thought there would actually be more. Turns out there's a lot less. And our hypothesis for this is that perhaps they're migrating out of the tumor into the lymphatics. At least that's what we hope. If it's not that, we're not quite sure what this means. And also, surprisingly, there are fewer tissue-resident memory T cells within the tumor tissue. And again, was kind of the opposite of what we expected, but it's clearly significant. So perhaps it's a positive thing that we just can't explain right now. Looking at phenotyping inside of the spleen, when we look at the transferred T cells, we found a significant increase of T cells that had the IL-12 construct still on the surface. So a week out, the T cells are still expressing and retaining that IL-12 anchored to their surface in the spleen. Additionally, we saw that same increase of granzyme B in both the transferred cells and the endogenous T cells, as well as a decrease of IL-2 in the transferred cells. We don't know what this decrease of IL-2 means, but it continues to come up as a pattern. So there's clearly something significant about decreasing the IL-2, increasing the inflammatory cytokines, and there's a positive benefit to that combination. Looking at phenotyping in the blood, we found that there was the same increase of both granzyme B and interferon gamma in the endogenous T cells. That's really what we're most interested in. And we did a P15E tetramer staining. P15E is an epitope of an endogenous retrovirus that exists in the cancer cells. So the idea is if you can stay positive with the P15E tetramers, that's already indicative of antigen spreading, meaning that that epitope was identified by the endogenous immune system, and then those T cells proliferated. Looking at the ELISpot data, again, we found that our IL-12 replicon treatment gave us the most number of spots. That is, the endogenous T cells from that group were able to recognize the antigen-negative cancer cells the most, better than any other treatment group, including the transmembrane and the mRNA group. All right, and finally, we did a rechallenge model to see if these mice retained the immune memory against cancer cells. So we took all the cured mice that we had in our initial round and then re-inoculated them with the same antigen-positive cancer cell line. And very quickly, they suppressed the tumor growth, and all the control group mice had their tumors grow out. After a few weeks, we inoculated them again with an antigen-negative tumor cell line. So now any remaining CAR T cells have no effect on these cancer cells. But the endogenous immune system had built up strong enough of a memory to also suppress the antigen-negative cancer cells, showing complete survival from this. So, in summary, we designed a platform to transfect primary T cells with the self-replicating RNA. We identified the membrane-bound or anti-CD45-bound IL-12 to be potent, yet minimally toxic driver of antigen spreading when expressed by the CAR T cells. And then across two syngeneic solid tumor models, we verified the necessity for endogenous T cell recruitment to have long-term efficacy and protection against those solid tumors. So thank you very much, and I'll take any questions. Yes. Sure. Yeah, I moved kind of fast there. Yes, I can hear you. Sorry.
Shows the post-treatment, the tumor volume decrease in the case of the IL-12 and CD45 treatment.
Mm-hmm.
And I'm not really sure about in your previous detoxing data, is the IL-12 and CD45 is the lowest toxicity one compared to the others?
Okay, I think I follow. Let me just go back to the initial experiments you're talking about. So there was this one with all the variations. Is this the toxicity one you're talking about? Sorry, I got a lot of animations here. Here. Mm-hmm. Yeah. So the IL-12, relative to the other groups here, we could say has the most toxicity, perhaps. But what we see here is that the body weight doesn't actually drop per se that much. What's stunted here is that we're using young mice, so their natural weight patterns will be growing since they're only about 6 or 7 weeks old here. So naturally, they'll just continue to grow. What happened here is, yes, there was a little bit of toxicity that kind of stunted that growth for about a week, perhaps, but then they rebounded and back up. So there was never a weight drop, and certainly not a weight drop near the 15 to 20% threshold that people could generally consider too toxic. So we considered this to be safe, even though there was that slight suppression for sure.
Yeah, I think that the last part of the data showing that means so the IL-12 and the CD45, those CARs, that's the potential to train the nice endogenous immune T cells.
Yeah. Yes, exactly. In a way. So the idea is how this antigen spreading works is that somehow we need to kickstart the reaction of cancer cells dying and releasing their neoantigens that are inside of them. So really the IL-12, we think, just boosts the CAR T cells initially, and also, I guess, boosts the other cells around them because they're anchored on the cell membrane to give that initial kick of the cancer, and hopefully release the neoantigens into the system for the dendritic cells to pick up, bring to the lymph nodes, and then retrain the endogenous T cells to see those. Because generally, if the neoantigens are kind of just refined to the tumor area, it's hard for the endogenous T cells to get in there to see what's being presented by dendritic cells because of all the very immunosuppressive cytokines that are being released by the cancer, and the other barriers to entry into the tumor microenvironment.
Is that means that if the patients get those kind of treatment in the once,
Mm-hmm.
and then they might get a long-term benefit?
Yes, exactly. So yeah, the idea is that hopefully, at least for the same type of cancer, if it tries to come back with different mutations or for example, think about it in terms of a cancer vaccine. If you get a vaccine against a specific epitope or a set of epitopes, the idea is over time during the treatment, your endogenous immune system will start to recognize even more of those epitopes to help protect long-term against future mutations.
I think I have some of the questions about the general field is, I think the hardest part of CAR T, it's not just about techniques behind the engineering. It's also because of the nature of the cytokines and which they activate to multiple downstream targets, cells, receptors. And how could you control this in an ideal range? Basically, like you want the pros better than the cons.
I see. Yeah.
Yeah. Talk about the alpha and we compare about the function of that in the T cells, but in the reality, they start a reaction from the innate immune responsibility. Every cell they respond to those cytokine at the same time too. So
yeah.
I don't know.
Absolutely. That's a very valid concern. And like everyone's always concerned with CAR T cells about like neurotoxicity, CRS. So many things could go wrong with using cytokine therapies. And it's a very difficult question to answer because we don't really have a good method right now of trying to boost the activity of CAR T cells without cytokines. So while that still is being researched, we're kind of playing this balancing act of how can we use cytokines in a safe manner. It's a very difficult thing to do. And of course, mice are far more robust than humans are and can withstand a lot more. If we start sweating, like we complain and like need to get out of the hospital immediately, but mice are more enduring than that. So just because we got positive data here in mice does not mean it could transfer to humans and immediately be successful. And of course, we see that in clinical trials all the time. So yeah, the cytokine battle right now is a very difficult balance to make, and hopefully there are different ways that we can discover how to do this in the future. Thank you.
Okay.
Thank you, Hong. Hello, everybody. My name is Gang Liu. I know I'm the last speaker, but I don't feel bad because a lot of background information has been introduced. So I'll have much less burden. So I know everybody may be kind of tired. I will try to put my story simpler to share some of our thoughts and our work that is about how to get antibodies that with their binding affinities, bindings, and function that we want. So first, I want to thank our partner, our long-term partner, on behalf of my company, Biointron. They are our partner not just for this launch and learn. Also, they offer us a lot of material that once was hard to make. So because of their work, we now are able to raise a lot of antibody hits for some targets. Their products like VLPs, nanodies, a lot of material for protein complex VLP are working very well. Okay, so since we already have such excellent choices for us to raise antibody for targets that once was hard for us to do, so we will not just satisfy with getting binders. We want to get binders that has some binding bias, or I would say have some bindings that we want them to bind, and we want what to happen once they bind to that target. No. Sorry. Okay. So since Homing has already talked something about our company, so my talk will be focused on binding bias, and there's some drug examples will be put, and also I will talk something about what we can do at the early stage to get the biased antibody that we want them to have desired bindings, and what Biointron has done, or what Biointron can help you in your antibody discovery-based project. Homing has already mentioned this, so I just skip service facility and what we can help. Okay, sorry. Okay, so when we talk about antibody binding biases, actually every antibody has its own binding biases. The fact is we did not care about them much at the beginning, at the first stage, traditional antibody drugs. That's why we, in many cases, we got have imperfect effects, and also at the same time a lot of side effects that we don't want. So in the next generation antibody drugs, what we can do from the very early stage, I would say we should not just satisfy with the general binding of our hits to its targets. We also need to care more about when, where, how much, and how fast or how slowly, and why we want it to bind this target. Only by doing this can we make the so-called precision drug possible. So by saying biases, I would say biases for precision, or if we cannot make the precision possible, at least we need to know how it gonna happen when it's bind to its target. What kind of good thing, a bad thing it might happen, and how well we can manage them. Otherwise, we will be surprised by the binding. Okay, let me give a general introduction about antibody binding biases. Actually, antibody binding biases is not just simply where or what the bind site is about this antibody while it's binding to its target. Something like multiple layered. Say we can talk about the epitope specific, which is the site where the antibody binds to either to the liner or its special structure site. Say I list somewhere here is like a membrane proximal dismal epside for us to form like stable strong immune synapse or loose, or we want it to have an enhanced internalization of the receptor. Or we say some target they have multi-domains, domain 1, 2, 3, 4, 5. So we want some of them that just function for binding, as a docking site. Some of them maybe function as a function site. So you may want them to bind to either functional if you want, or just merely just binding site. Or as we know that in some diseases, the white type protein, they are truncated, or some PTM happen to them. In this case, we may want our antibody to bind to specifically to this kind of site or new epitope that's formed because of the happening of the disease. Or in some complex targets, say they have a lot of subunits, or they are multiple transmembrane. So in this case, we may want the antibody to be bound to its specific so-called allosteric conformation, because by doing those we may get the downstream effect that we want. Or some target, they are kind of complex, not just one protein. It's a complex formed by subunit like A, B, C. So we want the antibody to bind only when the complex is formed. So in this case, we have to do a lot of work to find the binder that we want. This just the epitope specific antibody binding biases. When we talk about the biases, actually there's also a second biases called target abundance biases, which means for some antibody, they may not bind to that target only when the target's their level reach some gate. Otherwise, there will be almost no binding. So a second case is like so-called low affinity, high avidity binding, which means as we know, the mAb antibody have two arms. So if just one arm binds to the target, so the binding might be kind of weak, I mean unstable. Only when both arm binds to the target, in this case, the binding will become much more stronger and stable. So this is good for some antibody drugs that target to some antigen where, for which they not just is expressed in disease cells, also in a lot of normal cells. So by doing this, we can spare the normal tissue just for our antibody drug to target the bad cells. And also one third is called cross-linking dependent binding, as shows in the middle image. So just monovalent binding, maybe if monovalent binding may not trigger anything unless bi or triple valent binding happens. And also sometimes we don't just care about how strong or how weak the binding is. We may also care about how fast, how slow the binding is, which is called binding dynamics. Like fast on, slow off. In this case, a stable, long-lasting binding will happen. This case, I would say, is good for some disease target binding. We want it a long-lasting, solid, strong binding. So in this case, the drug will have much better effect. Or slow on, slow off. So in this, somebody may not know this case, but it's sometimes it help because although it's a slow binding, but it's slow but steady. So as everybody knows, so for solid tumor, our antibody may not penetrate into the tumor very well because as its name shows, very solid, tough to penetrate. So in this case, you want your drug to bind gently, tender, slowly, steadily. So otherwise, the drug maybe just stick to the out space. And one third case is like fast on, fast off. I think many people know about this. This case kind of working for targets for antibody targeting to like the effector receptor, like CD3, CD8, CD28 from 1BB. So in this case, we don't want these killer cells to be functioning too strong or too early. Also, one more is context driven. This is now in the recent years that become very hot. Like as we know, in some disease inside it, like tumor, there are something that's not happening in our normal cells, like lower pH value or some MMP enzyme with very high level, or like hypoxia or higher concentration of ATP. So we want to rely on these triggers to let us drugs to function where we want to function. Okay, in the coming 5 minutes and some slides, I will with some drugs to show you what the binding biases is and how these biases steer the drug MOA, and finally they may help achieve some fact that we want. So this is the cetuximab that everybody should know well. This drug now has appeared in the market for many years. Its target is EGFR. So as everybody knows, there are 4 extracellular domains for this receptor. So cetuximab, I would say it has a distinguished or kind of better therapeutic effect compared to some other targeted therapies or some of other paired product. The reason may be many. But I would say one of them could be, is binding biases. So as shows here, cetuximab bind to domain 3. So thus it can directly block the ligand binding, and also its downstream signal. And also by binding to this site, it locked EGFR in active conformation. And also it induced faster and better internalization compared to its peer products. And the second example, actually, it target the same target EGFR. EGFR is so popular. I think everyone know because it plays so important role in our cell growth. I would say cell renew, like our skin and our digestion system. That's why once we found that EGFR or HER2, they have a higher level in tumor cells compared to normal cells, we target by either targeted drugs or therapeutic antibodies. But their effect also comes with the toxicity, because our cell renew is required, is important, not also for our normal tissue like skin, digestion system. So if this signaling pathway is inhibited a lot, so it may bring something bad. So as shows skin toxicity and some other organ toxicity. So here shows is an engager. According S2 is EGFR binding, and binding the domain is binding to a specific epitope, where this epitope just work as a docking site, which means binding and binding only. No downstream effect will be triggered. And you may wonder, so in this way, how this drug can function to kill its target cells? Actually, as I say, it's a engager, it's a bispecific. So as the blue arms show, there is a bivalent anti-CD16A arms, which will engage the NK cells. So with by doing this, so this antibody will kill its target cells. And the CD16A also bind to FC receptor domain that's different from FC. So as we know, the NK FC gamma receptor 3A is also shared by our IgG1. So and also inside our body, a lot of, I would say, much higher concentration of normal IgG compared to this drug. So this antibody is 16A binding site is different from our normal FC. So in this case, there will be low serum IgG competition. The third example will come from a research, it's early stage. It's a SSTR, a GPCR target. So as everybody know, GPCR, they are a trouble not just because they are complex structure, also because its downstream function may vary depending on where you bind it. So it have multiple ligands, multiple binding sites. So if you want to have an antibody that after binding to a GPCR and want the downstream signal happen to happen as you wish, you have to get an antibody hits that binds to where you want. So in this case, this antibody first is binding in a conformation specific manner, binding only to somewhere between as shows in the photo is between ECL and ACL2 and ECL3. So by binding to this site, so this GPCR's conformation is locked. So hence the downstream cell proliferation is suppressed. And something else that I think is more important is that this binding site does not interfere with the normal SSTR binding. So in this case, there will be no, less unwanted side effect, because our body itself still need the normal SSTR2 binding to function. If we block everything, we may kill the tumor cells, but we may feel something bad may also happen. So, okay, this is example that's showing us that is so-called low affinity, high avidity. It's a 2-to-1 format of a T cell engager. So as you can see, the tumor targets that bivalent targeting. The target is ECA, while the CD3 just the monovalent. So by this so-called low affinity, high avidity binding and biases, so this 2-to-1 format TCE can selectively bind to disease sites where there are much higher CEA than in normal. So as I said, if just one arm, the red one show, one arm only binding to CEA, it may be unstable binding and go. So binding and go. So that is unstable. But if both arm bind to CEA, this binding will become much stronger. So by doing this disease selectivity can be obtained. And the next one is binding of affinity, dynamic bias. As I said, we may merely care about binding affinity. We also, in many cases, and more cases, we care more about how fast, how slow, how long our antibody bind to its target, because it is found that even without similar KD, so the drug may not have a exact same effect with the different K on or K off. The reason, as we just said, maybe because like if antibodies with lower K arm and lower K off, it may bind slowly, but bind longer. So this is very good, I think, for solid tumor targeting. This should be the last example. It shows there is a PTME trigger sensitive binding biases. So as we mentioned, so inside the solid tumor, there's a lot of things that's unique from our normal cells, normal tissues, like some unique MMPs and lower pH values and a very higher concentration of ATPs and hypoxia. So if we can rely on these triggers to fulfill this targeted binding, that will be very helpful for our precision antibody drugs. So in this cases is a early stage study. They compared, lower pH value binding biases, normal, higher, found that the pH sensitive binding does help for us to fulfill the context dependent antibody targeting. Okay, since we have talked a lot about antibody binding biases. So now we can see this so-called biases is multiple layered. So now the question comes to, so what we can do at a very early stage, and also with some case studies, I will show you guys what Biointron has done in the early stage antibody like in antibody taste discovery stage, antibody lead engineering, and also in many cases we have to design biospecifics. So what kind of format we can design, choose, compare to finally get a steered MOA. The reason we bother to do so, actually, the reason is very simple, because no target is clean, right? For solid tumor, except for solid tumor, no target is clean, but we can do something to make our drug clean. Okay, the first part I will talk about at the very early stage, which is antibody hit discovery. So what we can do to get a bias binding. Say we may do some direct epitope or domain or conformation and target. We can do this as early as animal immunization with some specifically designed material, or after we get the library, either phage library or B cell library, we can do some targeted screening to get the antibody hits that we want. Or in some cases, we have to do some counter screening to remove the hits that we don't want. So let me show you some examples that Biointron has done. So the first one is kind of simple. So our customer, they want VHH antibody that will not just cross react to both human and cyno. They want the binder to bind to just domain 2. Maybe domain 2 is a functional one or very important. So we after animal immunization and phage library, and we did solid panning, liquid panning. We use full length target domain 1, 2, or 3. This fragment for positive, negative panning, screening, and finally we got as many as 14 hits that specifically bind to domain 14. And sometimes we also get some other hits that target domain 1 or 3, because our customers sometimes they want to compare the function, the binding behavior, functional behavior, if you bind to domain 1, 2, or 3. Before I move to the next cases, I will give you guys a quick look at our VH antibody discovery. So Homing has shared a lot. So I will just point out some of our key features. So as he said, we have in-house farms. So we have 2 farms. Now we have like as many as in total 400 animals, I see. And the second is that all of those from animal immunization, which library construction, panning, screening, expression, functional screening, all the way, they are all full in-house, all done by us. So it's well controlled, not just fast, well controlled, and also it's free to operation for you guys once delivered. And also we have very flexible, very open to how we should design the project according to targets and according to your needs. So which means it's a targeted specific solution. Now we can, let's say, do some antigen modification to get some specific domain epitopes, or we do this to overcome some immune tolerance, or we shift from adjuvant A to adjuvant B to get a better immune response, or we adjust our dose or those inside. Or sometimes we will also do some change like short intervals, 2 weeks versus 3 weeks, and also rounds. Sometimes maybe 3 rounds is enough. If you want hits that can cover all of the sites, but you don't want each hit may bind preferentially to one particular site. Or if in some other case you want very super strong hits, you don't care the diversity, maybe we can like finish as many as 5 rounds, 6 rounds shots. Yeah. Also, we are good at using antigens like protein, mRNA, DNA, cells, VLP, and more. And also, as for the targeting class, we are not just good at proteins, peptides, also good at protein complex and GP cells, this kind of multiple transmembrane proteins, and also some small molecules, non-protein antigens. Okay, so this is the case 2, also the VHH is a TNFR superfamily member, so like 4-1BB, OX40. So as you may know, for this kind of targets, we cannot just get binders because we want functional ones. And we not just want function, we may want blockers, non-blockers, and more. So positive, do some positive work or negative work. And also that's not everything. We want it to be ligand dependent or ligand independent or ligand competing or ligand incompeting. So a lot of biases, a lot of choices that we all want, because for targets like 4-1BBs, as everybody know, is a very good target, very valuable, but it comes as the monospecific, comes with severe hepatotoxicity. So till now, we still have not yet figured out exactly what we should do to get a good monospecific antibody targeting 4-1BB with much little side effect. So we have to have a panel of hits for us to to compare design. So in this case, you may not just want one type of hits, you may want a lot. So as shows here, we can have hits with variable binding behaviors, as shows in the SPR charts, like fast on, fast off, slow on, slow off, or fast on, slow off, or slow on, fast off. So because for this kind of targets, as we said, just as in the story of CD3, you don't want the target to be the binding to be too strong or too fast or too solid. So, but you don't exactly know which one is the one that fits your needs. In this case, you have to have plenty of these ones for you to choose and compare. And also the same thing is true for its function. We want blockers, non-blockers, agonist, and ligand competing, ligand non-competing, a lot to choose. So we are able to get all of these. The next study is biased anti-GPCR as mouse antibody. So as we have already know, GPCR is a target, hard to get enough hits for us to compare, and hard to get the one with the biased binding and biased function. So in this case, we use mRNA to immunize our mouse, and then with some engineered cell line, we got our biased blockers. So you may wonder, how can we get a mouse from alpaca? Actually, this mouse is raised not from alpaca, it's from our AppDrop single B antibody discovery platform. Tommy has talked a lot about this, so just want to know that it's a microfluidic drop driven and droplet-based single B antibody screening platform. I'll skip this, and the second. I still want to point out some of our key features of our AppDrop. First, so as Homie also mentioned, it's a fast track. So from for the B cell screening, just 1 day. From cell to paired sequences, just 1 week. From sequences to antibody, 2 weeks. So in general, from shots to hits, just 2 to 3 months. So very fast. So it's not just speed, it's also one other thing many people care more about is antibody diversity. So since we are mining as many B cells as possible, so we are having much big chance to get B cells, positive B cells, and hence get much more diverse sequences. This is very good for when you have some challenging targets which may trigger very weak immune response. So in this case, if you lose plenty of B cell clones, you don't have many left for you to get sequences. And also, as the coming slide will show, we're not just good at the mouse, we're also good at a rabbit, and more. So in case like you want anti-mouse antibody, I would say better not to get it from mouse because the immune response may be zero or very, very weak. So in this case, you may choose rat or rabbit. Or if you want antibody that has very high affinity, you may choose rabbit. And also, if you say you want to antibody that's very good yield, you may choose rat. Yeah, so something like this. So this case shows anti-small peptide mouse antibody. So this peptide just 45 amino acids long, very low immunogenicity because its structure is so small, so simple. So still we got blockers by design cell line based selection. So the part 2 will be what we can do during at the stage of antibody lead. What kind of mutations, engineering work we can do to tune the binding for some specific contexts and it's still at MOA. So we may tune antibody affinity as we just showed, low affinity, high avidity, or we may engineer its K on K off rate to get the binding behavior that we want, or we get some conditional activation like in the low pH value or high ATP concentration, or if in some cases we have to use some specific long CDR3 loop to target some epitope that's kind of big but smooth, which is very hard for general antibody to target and bind. We may have to do some engineering for these CD3 loops. Here comes some examples to show what Biointron has done for antibody release engineering. First is the VHH antibody affinity maturation. I know many people like VHH a lot because they are easy to produce, smaller size, and some other CMC good for CMC traits. But there's something I would say kind of not that great, which is their affinity. So in some cases, we have to do something extra after we got his. So in this case, you can show this parental VHH give us a lot of room for us to increase its affinity. Shows in the 3 mutants, we increased its affinity by more than 1,000 times, 300 times, 700 times. So it's very great. So you may wonder how can we increase its affinity by more than 1,000 times? That's because of our site saturation fully covered antibody affinity platform, which means for each CDR site, for each site as each of any other 17 amino acids, we mutant to that site, to that possibility. So every CDR site, every possibility is covered. So by doing this, we screen the whole library. Finally, we got the hotspots. This is what happened in round 1. In round 2, we may combine some hotspots and then go to round 2 to get the final hits. And this is not good, not just for affinity maturation. Anything that you want to do about its affinity, like their binding rate, like context sensitive binding, we may try to do this. And also one more thing I want to mention that this platform, we express all of those variants with chill, so no fetch. So that means no PTM gap. In the second example shows while we remain on rate, which is already very high, very good, our customer wants a slowed off rate. So we can just focus on K off. So as shown in this slides, retain K on, but much improved K off. And a similar story in the case 7, but it's to keep K off rate, but increase the K on rate. And the last is the pH value biased. So we can also do that. So if you want to get some pH sensitive hits, we can use our site saturation platform to do so. But we will not mutate each site to all of the 17 amino acids, just to his. And also screening the his, and then we combine some hotspots, and then we screen the his candidates in lower pH value and normal pH value to get a pH preferred binding. This shows example about we can increase the pH sensitivity to like the gap, the difference can reach as high as 18-fold. The last will be the drug format. Okay, actually 3 minutes. So I know, some in many cases even though we have very good sequences, but if the format is bad, that cannot help. That may hurt our MOA, drug function. So in this case, we have to do something like prodrug or TME sensitive or avidity driven or split functionality and many more. So I would say Biointron may not help you much in how to design because that's your idea. Like say trispecific, bispecific, 2-to-1 or 1-to-1 or T-cell engager, NK engager, low affinity, bivalent. It's up to you. But we can help you if you have some great idea. If your targets are fixed, but you don't know exactly which format will be the best with the function, behavior, binding behavior, binding biases, function biases, and also very good CMC traits, we can give you, prepare those choices. So when we give you all of those samples, you can test by yourself. The reason why we feel so confident is, as Homing said, we are very good at antibody production, not just monospecific, also bispecific, very fast. In 3 weeks, with these highly automatic workstations, we can finish our even bispecific antibody production in 2 to 3 weeks, very fast. And also not just fast, and also with scale. Because we don't use stable cell line, we just use transient transfection, which will save a lot of time. And also have rich past experience, as shown have finished 40,000 bispecific production. And with 400 different formats, we have very, very rich past experience. This just show you some cases that we finished like symmetrical, asymmetrical, Fab2 SLV, Fab2 Fab, 2-to-1, 2-to-2, Fab2 some ECD with linker or without linker, with 2 links or no links for, or tandem or not. So a lot of format that we can, if you want any idea, we can test. So I'm gonna end my talk by this study. It was just published last week, actually a few days ago. It's a PROMAC, but from this case we can see how binding biases, format design, and conditional assembling help the functional precision happens. First, this product is a prodrug, but it is compared to split with each prodrug. Why is prodrug? The prodrug is made possible because of the tuned repulsive charge pairs. As you can see, the two paths linked by a linker in the prodrug A and in prodrug B. So they're both negative charge and positive charge. But the charge could not be too strong, otherwise there will be no binding, or could not be too weak. Too strong, there will be no something the reassembly will not happen. But if the charge is too strong, there will no binding. So that's why I said the repulsive charge is well tuned. So although in each of the prodrug, the binding still happens, but it has no function. But when both prodrug reach the same cell, must be the same cell surface, So the targeting arm will bind to both. Then the binding will be stabilized. At the same time, the CD3 targeting arm will resemble habit. So as you can see, negative charge, positive charge, they resemble. Thus, the functional CD3 binding arm now is formed. So by then, this bispecific, T-cell engager now begin to function only at the site that we want. So that's called target precision. Okay, so put simple, as we now always say, precision begins with bias. So if you have not yet partnered with Biointron to steal function from antibody discovery. Thank you.
All right, thank you, Dr. Liu, for for everything. And at the end, we're gonna draw the final prize with the Apple Watch, right?